So we will now proceed to the last presentation for this session. So let me now call on Dr. Vanessa Balois to start.
Okay. All right. I hope you can all see my slides. So good morning and thank you very much for the opportunity to present in this online conference, especially PAASE and also Dr. Almoro for extending this invitation to me. And a little bit of trivia, I recently got married. So my
Yay!
my actually new surname is Oguchi, but for now I'm still Maria Vanessa Balois. But in after the documents are all finalized, it's going to change soon. But anyway,
Woohoo!
thank you for the kind introduction, and I'd like to give a presentation wherein we show the development of near-field scanning optical microscope systems for nanomaterial characterization. And I am affiliated with RIKEN, which is a research institute here in Japan. Now, to give a short introduction, what is my team? What is the Innovative Photon Manipulation Research Team? So our team focuses on creating sub-wavelength structures which we make and use to observe or measure certain properties of our material or of the sample material. And our research is basically split into two kinds of research. The first research is by making these and designing these structures and then using them for practical applications. And we call these sub-wavelength structures metamaterials. And one of the applications of our lab right now are mainly absorbers, sensors, and also surfaces for light manipulation. And a little bit of trivia, some of you may see a familiar face here in my slide. We have one Filipino student from UP, Dr. Hermosa's student, Miss Cherry May Olaya, and she's currently finishing or doing her PhD research in our lab here. Now, the second aspect of our research is that we make these sub-wavelength structures and then we use them to observe near-field light matter interactions. And for these light matter interactions, one of our group mates or lab mates is studying chirality, while as for me, I study or I use a sharp metallic tip, which I use to scan and also observe the very local physical chemical properties with resolutions beyond the diffraction limit. And this technique is called near field scanning optical microscopy. And our team is headed by Dr. Takuo Tanaka, who is an expert in both metamaterials and also plasmonics. Now, going to the meat of the presentation, I mentioned we are using a technique called near-field scanning optical microscopy. Now, there are a variety of, so it's a big family, and one particular member of this family I'm using is TipEnhanced Raman spectroscopy. For those of you who are not familiar, TipEnhanced Raman spectroscopy combines a tip or a plasmonic metal tip, wherein when you shine laser light on that tip, a nano-confined light source is generated at the tip end due to photon confinement and leads to a tip-enhanced electric field. And combining this tip with what we call Raman spectroscopy, which is a very powerful technique to distinguish the chemical properties of a sample, that if you image each single peak from the Raman spectrum, you can actually get various kinds of information. Basically, Raman spectroscopy is done in the far field wherein you do a large scan of the sample. Here in this image, it's actually 10 microns by 10 microns. But for tip-enhanced Raman spectroscopy, we are actually interested only in 1 pixel of that entire image. And then in that 1 pixel, we want to image that small pixel to get the very local properties of our sample, which is why TERS has been dubbed that it is an analytical technique with both high chemical sensitivity and also high spatial resolution. Now, in our team, we want to not only develop, well, the main goal is to develop the system. So there are three factors which we want to develop, but there is one core idea behind everything. It is simply making the invisible visible, which is actually the main purpose of a microscope. And in our terms, we can think of it, instead of a microscope, it's a nanoscope, because you want to see nano-sized materials. Now, the three important aspects we are trying to tackle now are: first, chemical sensitivity; second, spatial resolution. And once we have these two, we want to make our results repeatable and reproducible, which is why we also emphasize on system stability. Now, the flow of this presentation is, I will be first focusing on chemical sensitivity and how it goes along with the system stability for one particular application which I developed during my PhD, which is tip-enhanced terahertz Raman spectroscopy. And the other half of the presentation is dealing with high spatial resolution, which in turn, because of the high system stability, can also enhance the chemical sensitivity of the entire system. So let's dive into everything else. Now, tip-enhanced Raman spectroscopy is a very great technique, but the main limitation is it is limited only to the fingerprint region. Fingerprint region means basically the 200 to 2000 wave number of the Raman spectrum, if you look at the spectra. Now, the interesting part is, so far, the lower frequency regime, which we call terahertz Raman. Please do not confuse this with the terahertz wavelength, which is in the far infrared. So that's totally different. Now, here, terahertz simply means low frequency, and this is basically from 5 wave number to around 200 wave number. So these frequencies are near the Rayleigh line or excitation line. And here we can simultaneously detect both the Stokes and anti-Stokes Raman. And these aspects I will show you in this slide. So the advantages of using tip-enhanced terahertz Raman spectroscopy and also the terahertz or low frequency Raman that you can detect are, for 2D nanomaterials, which are layered, they're very useful to find out the number of layers of your sample, for example, graphene or molybdenum disulfide. And then another aspect which is interesting is that using these low frequency Raman modes for carbon nanotubes, you can determine the diameter and also the chirality based on what we call the radial breathing mode. Now, I think for me, the most interesting part for low frequency Raman is in the biological applications, particularly for medicine. Now, take for example carbamazepine, and if you look at the fingerprint region or the 200 to 2000 wave number, if you compare form 2 and form 3, two kinds of carbamazepine, the fingerprint region or the Raman signature is totally identical. So it's very difficult that you don't know, based from this fingerprint region Raman signal, if it's a form 2 or a form 3. But if you look at the low frequency, they are totally different. So here you can clearly determine which carbamazepine is in the form 2 and which carbamazepine is in the form 3. Now, the research which which I tackled, is mainly about the problem of heating because of the enhanced electric field that is generated at the tip end. And how can we extract this nanoscale or nano volume temperature and use it for practical purposes? Now, low frequency Raman is useful for temperature determination because you can actually calculate the temperature based on the Boltzmann distribution and also the intensity ratio of both the anti-Stokes and the Stokes Raman. And by doing it via experiment, obtaining the anti-Stokes and the Stokes intensities and then crunching out the numbers based on this equation, you can actually calculate the temperature at the tip end, which is more or less the temperature at the tip, at the sample. Now, the setup we used is a conventional atomic force microscope system where we have a tip, which is either gold coated or silver coated, and depending on your laser illumination. And then the special part of our sample, or not sample, but our setup, is we use special notch filters which filter out the Rayleigh signal and make it possible to detect both the Stokes and anti-Stokes frequencies simultaneously. Now, as a result, we were able to calculate or to extract the anti-Stokes and Stokes Raman from the carbon nanotube. And from there, we were able to detect sample temperatures less than 100 degrees Celsius. Now, one thing I have to point out here, since I was so excited about the temperature, is please look at this image over here, which is the TERS image and also the topography image. Now, the other powerful thing about TERS is that you can get both the chemical information and the topographical information simultaneously. And note here that this is actually two carbon nanotubes, and I'm sorry, but the uh, scale bar here is around 100 nano. So we can actually see very, very thin carbon nanotubes, both in the TERS image, which is the Raman imaging of the G band, and also the topography. And lastly, going back to the temperature, we found that you can control the temperature, well, low temperatures less than 100 degrees Celsius by simply controlling the input power. And with this, we found that this is very much an exciting topic to pursue. And right now, we are trying to improve the detection sensitivity of low frequency Raman by also increasing the number of filters or notch filters in our system so that we can have a better signal to noise ratio. So here you can clearly see from the 2-notch filters to the 4-notch filters, particularly at the center, which is close to the Rayleigh line at 0 wave number, the peaks, especially in the anti-Stokes, can be more clear, well, can be clearly seen for the 4-notch filters that were used. And aside from that, uh, for of course, we want to have better chemical detection by improving the instrumentation, but we also want to have the system to be more stable to do better or even long-term uh, measurements. Now, we also have a temperature booth installed wherein it controls the temperature, and the error is around plus minus zero point 0.1 degrees. So the red line is inside the booth and the blue line is outside the booth. And I would like to personally thank Dr. Desalias because he was also very instrumental in improving the condition of this temperature booth during his stay as an intern in the Nanophotonics Research Laboratory. And we found that we were able to create a very nice, stable temperature environment to prevent what we call thermal drift. And another factor which we want to consider so that we can also shift to what we call long measurement times is that we want to ensure that the focus, the laser focus on the sample is always at focus. And to do this, we have implemented this system wherein we use a capacitive sensor, which helps maintain the focus spot position of our objective lens. And the stability of this system is plus minus 1 nanometer. Now, to clearly see how this autofocusing system works, let me show you this very nice slide. Without the high stability system, you can see that as the time increases, the temperature of the room also increases, and that also causes some defocusing because of the change in the environment. And over time, at first, at the start of the measurement, you still have a more or less focused image, but as the time goes by, it becomes very much defocused that you cannot anymore identify what kind of structures you are trying to make or to image. But with the high stability system, you can clearly see these strained silicon nanowires, which are nicely spaced from each other. But so far, the system is only working for what we call conventional microscope, which is just far field measurements, wherein there is no tip involved. So we want to integrate these two systems together as a next step. Now, the other aspect of the system is a while ago I mentioned an atomic force microscope, and now we also have another system which is called a scanning tunneling microscope. So they're both part of the scanning probe microscope family. However, the mechanisms are quite different. And unfortunately, I do not have enough time to differentiate both of them, but we use both of these techniques for our TERS experiments. Now, the STM TERS system is also, we improved the environment by putting our system inside a metallic environment control chamber, wherein, as you can clearly see here, inside the chamber, the temperature is very much stable and the humidity does not vary much. The humidity varies only maybe plus minus 1 degree RH, 1% RH. But outside the chamber, you can see the very dramatic change of the temperature, which is very not good for our system. And we also, this system can also be compatible with well, nitrogen. Like you want to purge nitrogen into the system, and when nitrogen is purged into the system, the humidity actually goes to zero, which is very, very nice for samples which are very much uh, well, sensitive to the atmosphere, particularly highly oxidizing samples. Now, let me shift from the AFM to the STM. Now, the beauty of the STM is that you can control very well the tip sample distance, which means that the stability is very nice. And using this system, we were able to get a very high spatial resolution of zero point 0.7 nanometers. And this is currently the world's highest spatial resolution for ambient TERS. Now, the sample that we studied is simply a gold uh, substrate with gold one-on-one substrate with a monolayer graphene, which is pristine, wherein there are no defects. And we scanned a large area here of 50 microns by 30 microns, but the actual scanned area is just this small area over here. Now, the very nice thing about not only having high spatial resolution is we found that if you have highly confined light, you can alter the Raman selection rules. And this means that you can maybe change even the chemical sensitivity of your system. And this was shown using surface enhanced Raman spectroscopy, which is simply, well, it's diffraction limited. So the results in our by our collaborator showed similar work wherein they were able to observe what we call forbidden modes, which are associated with defects on graphene, but their sample did not have any defects. It's a pristine sample, but they were able to still observe these peaks. And the main question is why? And we think that this is because of the highly confined field that decays very quickly. And this is a field gradient, which in turn also changes the Raman selection rules, which means that we indeed are now making the invisible visible because phonon modes which are not supposed to exist are starting to show themselves, particularly these defect related peaks, which are not supposed to be there because our sample is defect free. And we think that this is due to the non-vertical optical transitions, which are caused by our near-field system, which causes the well causes it to actually activate these forbidden modes and also detect them. And the nice thing about this is, so okay, we have high resolution system. We can actually also do imaging because we can also see the topography. And we found that we can also observe these nanoscale strain variations. Note that this image is only actually 23 nanometers by 14 nanometers in height. And we found that these nanoscale strain variations can be directly observed. But currently, they could not be directly observed because of the diffraction limit, and they can only observe it via the broadening of certain Raman peaks. But now, using our system, using TERS, we can now also do such kinds of high resolution imaging and also obtain very small information with high chemical sensitivity. Okay, so in a nutshell, so to summarize my presentation, the main goal of our team is to use sub-wavelength nanostructures to observe and also see certain things from our sample. And basically, we want to make the invisible visible. And this is possible by using a technique, well, in my case, called the Nearfield Scanning Optical Microscope, wherein we use a very nice technique called TipEnhanced Raman spectroscopy and TipEnhanced terahertz Raman spectroscopy for high chemical sensitivity. And so far, using the STM, we are able to obtain the sub-nanometer spatial resolution, which in turn can unlock certain Raman modes, which can in turn also increase the chemical sensitivity of our system. But all of this, or these two, of course, should go hand in hand with high system stability. Without the high system stability, we cannot get reproducible and reliable results. So thank you very much for listening to my presentation. And to end my presentation, I would like to acknowledge my research collaborators, both in the Surface and Interface Science Laboratory, and also particularly Dr. Francesca Celine Catalan, who helped in the work for the TERS temperature measurements for tip-enhanced terahertz Raman spectroscopy, and also Professor Riichiro Saito of Tohoku University for the very insightful discussions regarding the transitions of the forbidden modes for near field due to near field excitation. And I would like to thank these funding agencies for their financial support, because without the financial support, the progress of research will be very much slower than it is. So thank you very much, and I welcome your questions.
Thank you, Vani, for the nice presentation. We have time for maybe one or two questions. So we have a question from the chat box from Mr. Raymond Birtudaso.
Mhm.
What do you think the instability occur during analysis?
During analysis,
Uh,
so I would say that uh, analysis, as in processing the data, if that's my understanding, for data analysis, we usually take into consideration the errors due to the experimental system, and these errors are very much accounted for. We know the for the spectrometer, we actually know the uh, um, error margin for the measured Raman peaks, and also we we also do some filtering also for the imaging, to make sure that the signal to noise ratio is also suitable and m- gives a clear image of the image data. So I'm not sure if that clearly answers his question, or if that's the gist of his question.
okay, thank you, Vani.
Hmm.
We have a second question from Dr. Dasalias.
Yes.
Hello, Atibani. Thank you
Hi,
for the
Lian.
exciting presentation again.
Hmm.
I'm just wondering, is it a conscious effort to make the temperature lower in your measurement, or higher temperature is better?
Okay, so that's a very good question. So currently, my research is focused on room temperature. So we're just dancing around the values of around 19 degrees to around
Uh,
23 degrees.
I'm talking about the temperature on the enhanced field at the tip.
Okay. So yes, the I guess the key point for the temperature underneath the tip is for temperature sensitive samples, particularly biological samples, the lower temperature is better, because of the you can damage the surface, let's say, of a cell or of a molecule, and then, or you can also actually inadvertently cause chemical reactions simply by applying heat. But for more robust samples, which, so for example, graphene or maybe silicon, as long as the heat temperature does not cause a Raman shift due to heating, that is still okay. So if you, because for Raman spectroscopy, we usually observe the shift of the Raman shift. And this shift can be caused by strain, by interaction with another molecule, etc. But we want to make sure that shift is simply due to the interaction or due to the strain experienced by the sample, and not due to the heating, which can also cause shifting. So for those kinds of experiments, we still have to do a power Raman shift calibration to make sure that the Raman shift is still constant at a certain power. But once it starts this shifting because of higher power, that's when we say, okay, this power is not good. So that heating caused by the tip is also not good for the experiment. So yes, I hope that answers your question.
All right. So, Bani, marami kang fans. So, for the last, we have Dr. Tarun.
Ah, Sir Al, hello.
Hello, Vani.
Long time.
Congratulations.
Thank you, sir.
Wow, it's been a while.
Yes, sir.
Magtanong ka na.
Ang tanong ko ay, so in the resolution of 0.7,
Yes.
do you know what is the distance between the tip and the sample?
So right now, a quick answer to your question is no, but we are assuming that it's around 1 nanometer. But we
So
haven't.
is there a correlation between the tip distance and the spatial resolution?
Actually, that's a very good question. That's the next step of the research. So right now we're trying to further improve the stability of the system so we can do some tip sample distance and observe the evolution of the Raman peaks by increasing yes. Thank you very much.
Okay.
So, thank you very much,